First, let me thank you for the kind invitation. I debated as to how to begin my talk with you. And fortunately, I had an insight as a consequence of the fact that I'm old. And so I had to come a few days early to acclimatize, and I decided to visit the environs of Dubai by taking the metro. And on one of my first excursions on the metro, it was exceedingly crowded. And so I stood. And at the other end of the railway car that I was in, there was a middle-aged gentleman who motioned for me to come and take his seat. And I, of course, said, no, I can stand. I nodded, thanking him, but persisted in my spot standing. And when I got through with that, another gentleman on the other side of the car got up, had somebody hover over the empty seat, and he came and took me to his seat. So I sat down and, of course, thanked him. And as I was then getting off the train, the original gentleman who nodded at me came over to me and said, "Isn't it nice to see how many kind people there are, and that you thanked the gentleman by accepting his kind offer when he made you sit down?" And with that I had a vision, and that is how differently we see the world from our own orbit. On the one hand, the kindness of this person, and my thanking him. On the other, why I initially resisted efforts to sit down. Namely, I knew I was old, but not that old. So with that as the background, I want to talk to you a little bit about the marvelous technological advances and complexities of genetics, and on the other hand, remind you of the old paradigm of physical diagnosis and the art of medicine. And so you see before you some cells that we derived, neuronal cultures obtained by making iPS cells from fibroblasts from children with autism. And on the other hand, I want to point out to you that I am but a spokesman for this last group of delightful young people who do all the work, or who did all the work, that is somehow attributed to me. Okay? So I want to first begin by highlighting for you the observations of a clinician from 1943, a psychiatrist by the name of Leo Kanner, when he first described 11 boys who had a disorder subsequently known as autism. And he pointed out several features from their early infancy. The first, the absence of the usual anticipatory response that a newborn exhibits to its mother, to anyone approaching it. The second, that as they grow older, they appear to be remote from affective and communicative contact with people, and they develop a remarkable relationship to the inanimate environment. And the third, they are so concerned and enamored of the external world that they attempt to have it remain static, namely their stereotyped behavior. And this then was the original description of a behavioral complex, subsequently that came to be known as autism by Leo Kanner. The second, a different set of perspectives. An article that appeared a year ago in the Wall Street Journal about a young man with autism, ostensibly high-functioning autism, who won a Rhodes Scholarship. The observations of his mother, that these children may have amazing minds, and that all it takes is to be able to develop communication with them, to unlock their skills. The second, by the young man who was going to study ornithology, and he was asked why he was so preoccupied with birds. And he mentioned that they are disconnected from their surroundings, but they can escape their immediate surroundings by flying away, something he would like to do. And yet a third set of observations, the one I'll highlight is the observation by the Oklahoman American humorist Will Rogers. You know, everybody's ignorant just on different subjects. And finally, a quotation by the author of House Rules, Jacob's Room, ostensibly a young man with autism, is a place that entropy goes to die. It mandates order and structure and unchanging. So what then is autism? I want to suggest to you that autism, as originally described, is a behavioral trait, a behavioral characteristics, that it has been enlarged into a group of disorders known as autism spectrum disorders, and that what all these different disorders share in common is a lack of reciprocal social interaction and limited verbal and nonverbal communication. It is, however, a disorder in the United States that has been made or has become recognized as becoming exceedingly common, occurring in 1 to 2% of the childhood population, that it is a disease that is more prevalent in boys than in girls, speaking to its genetic etiology, but finally to remind you that what was originally described is a behavioral constellation. And I would put with you the following questions. What are the phenotypes and genotypes of autism? And stress to you the great importance of phenotypic characterization. How do we make the diagnosis, and how do we study its pathogenesis and pathophysiology? Starting again with the view that it is a behavioral trait that coexists with other neurologic symptoms and diseases, as exemplified on this slide, that its etiology is complex, multifaceted, that includes neurological, genetic, and environmental factors. But again, to stress to you its heritability. It is the most common of neuropsychiatric disorders. In monozygotic twins, its concordance has been identified as 70 to 90%, that the recurrence risk in siblings is 20 to 40 times the general population. As we've come to know with the advent of more and more research, that there are hundreds of loci and genes involved in its pathophysiology, that it has a male to female preponderance, and something most forgotten by clinicians, that 25% of siblings of children with autism will have major speech delay. And I stress that to you again because of identifying the phenotypic variants of this disorder. We tend to establish the diagnosis based upon neuropsychological assessments, on the one hand of the parents with quantitative and subjective various tests, and finally in children by doing neuropsychological tests that ostensibly give us quantitative data. The clinician, on the other hand, has to stress the birth history and family history, the developmental history of the affected patient, must do a very thorough phenotypic or physical exam, and that as time goes on, we have variable, more and more sophisticated laboratory methodologies for establishing a potential etiologic diagnosis. In the olden days, before I even had the privilege of meeting some of the younger members of this group, we did very unsophisticated diagnoses. We did karyotypes. And then the karyotypic or chromosomal analysis became more sophisticated, and we did high-resolution bandings that allowed us the first glimpse of, if you will, phenotypic chromosomal variation, deletion, duplication, and 3 chromosomes. And 3 loci on those chromosomes were identified as having a relationship to the pathophysiology of autism. I want to stress to you that those early observations identified loci on those chromosomes for neurodevelopmental genes, as well as the potential function of non-coding RNAs in the pathophysiology of autism spectrum disorders. With the development of comparative genomic hybridization and microarrays, we've come to recognize a host of additional chromosomes and loci in which either deletion or duplication is identified as causal in the pathophysiology of this group of disorders. With the most contemporary chips at the present time, it is now possible to identify over 2,300 copy number variations that play a role in the pathophysiology of autism spectrum disorders, and the recognition that we may identify such findings in 10 to 15% of patients who have the diagnosis. What is more important are the generalizations that come from those observations. First, that individuals with autism spectrum disorder have 10 to 20 times the number of copy number variations as compared to controls, that the greater number of copy number variations is associated with intellectual disability, and that larger copy number variations are found in affected females as contrasted to affected males, suggesting that females may be more tolerant to the genes that give rise to its pathophysiology. And finally, that the identification of genes is insufficient to define the mechanisms that give rise to these disorders, and that fine regulation of gene disorders or of gene dosage may play a prominent role. I want to stress once more that 10 to 20% of individuals with autism spectrum disorders may have a copy number variation that contributes to the phenotype, but that when one looks at these deletions and duplications, 3 particular network functions become apparent, specifically neurotransmission, synapse formation, and ubiquitination as being biological processes that may give rise to this syndrome. What do these newest technological tools, such as the ones you've heard about earlier this morning, add to our understanding of this syndrome? First, that de novo mutations are found in individuals and individual families that appear to be associated with the diagnosis. Secondly, that these mutations support a de novo paradigm as giving rise to disrupted pathways which give rise to this neurodevelopmental disease. That the diagnosis of exome sequencing, be it whole genome or whole exome sequencing, coupled with microarrays as diagnostic tools, enhances the number of individuals in whom one can identify a specific pathophysiology. And finally, there are biological processes that appear to play a role in the pathophysiology, such as neurite outgrowth, which one can examine in a variety of different systems. If you look at the present autism gene databases, one now recognizes that there are more than 800 genes that have been identified with the pathophysiology of autism. Clearly, single genomic or single gene mutations, such as you heard about earlier this morning, obvious ones with a symptom complex that the clinician tends to at least highly suspect and recognize. It identifies for us that there are two categories that shift as the years go on, namely syndromic and non-syndromic autism. That you can begin to assess in a less expensive way using microarrays targeted for certain phenotypic components, such as intellectual disability and autism, seizures and autism, that give rise to more rapid diagnostic methods, such as this particular example of a chip that allows you to diagnose some 2,000 genes that have been shown to be associated with the diagnosis of autism spectrum disorders. What comes from all of these sequencing data as of today is that one can identify candidate genes in an additional 5 to 10% of cases, that the genes are private mutations occurring within a given family, that a large number of these genes give rise to a risk for this neurodevelopmental disorder, and that most of these genes are involved in normal brain development. If you in fact couple array data that looks at copy number variation with sequencing data, you can arrive at potential pathways that must be fundamental to the defect in autism spectrum disorders, such as synapse formation and function, neurotranscription, and translational regulation. How do we conceptualize these findings in the context of brain development? And you've heard again about examples ranging from transcript variation or splice variation, alteration in the number of expressed genes, increase in the abundance of a variety of non-coding RNAs, but also to pay attention to brain transcripts or transcripts of genes that are specific to brain development and function, but recognizing also that there is anatomic or locational variation with regard to all of these phenomena. And that these phenomena play a role not just in terms of the anatomy and embryology of the brain, but also have a temporal pattern superimposed on them, so that various processes are more important in the latter half, for example, of pregnancy and the first few years of life, and that those potentially have something to do with the mechanisms that give rise to autism spectrum disorders. As an example, some work from our own lab looked at the difference in genes expressed in the brain in different regions of the brain, such as those that have something to do with autism, the prefrontal cortex, the cerebellum, and that what you find if you look, for example, at long non-coding RNAs, the difference is greater in terms of expression of those between the prefrontal cortex and the cerebellum in autism, and is greater in controls as well than is the difference between autism and controls in terms of long non-coding RNAs that are expressed. So the question becomes, do we have model systems with the ability to examine the pathophysiology of these events? And we all know about animal models and their limitations. I want to stress for you a set of models that is becoming an ever greater tool as we go forward. Specifically, that of inducible stem cells derived from mature cells. This is an example from our lab of the formation of neural cultures derived from skin fibroblasts that have been converted into inducible pluripotent stem cells, but to remind you of what may be a strength and a weakness at the same time. The neural cultures that we derive are not pure neurons. They are mixed cultures of glial and neuronal elements, and there is variation in the percentage of glial to neural elements based upon the procedures that are used, as well as based upon the cell of origin of the iPS cells and the neuronal cultures that you subsequently obtain. What is important, however, is that these neural cultures derived from mature cells can be shown to exhibit electrophysiologic processes that are fundamental to the brain and central nervous system in situ, and that in fact when you compare a small group in our hands of cells derived from boys with autism as contrasted to their normal brothers who have no neurobehavioral diagnosis, as contrasted to boys from the general population, it appears that there may be differences in central electrophysiological elements, and the ones shown here are difference in the type A potassium voltage and in the sodium voltage-gated current when you contrast autism-derived cells from unaffected siblings from controls, and that this may be an uncovering of a fundamental function that is not specific per se for autism, but is dysregulated in cells derived from these individuals. Furthermore, that if you look at gene expression in these cultured neural systems, you will find in our hands 160 or so unique genes that are exhibited in significant increase, that the genes have identifiable chromosomal loci that have been associated with the pathophysiology of autism, that if you look at various databases, you will find that the genes that are overexpressed have a relationship to things such as attention deficit disorder, schizophrenia, bipolar disease, that they're involved in synaptic transmission and adhesion protein biology in the central nervous system, and that at least a third of these genes have been specifically identified as having a fundamental role in the pathophysiology of autism. And when you do network analysis, you can identify at least 3 gross pathways that these genes have a role in, specifically synaptic and neural adhesion components, in transcriptional regulation, and in RNA processing. Finally, the brain is a complex organ. It has roughly 10 to the 12th neurons with over 10 to the 15th connections as we become mature. The question is, how do we make this give us an answer as to the specific etiology of either the behavioral trait or the diseases that are a part of the autism spectrum diagnosis? And I would like to end with a quote from the Nobel laureate Walter Gilbert, who wrote, "We haven't been able to determine in genes of what makes a human being a human and not another mammal. Consider the human brain, a collection of 86 times 10 to the 9th neurons with trillions of connections, yet it orchestrates everything from understanding, memory, to movement and sleep." So the objective of this conference and of my brief talk has been to suggest we're on the threshold, but not yet there, of personalized or precision psychiatry, that our hopes are based upon the importance of good physical examination and the definition of phenotypic diversity, that we need to employ ever increasingly and with greater sophistication the forefront diagnostics tools that we now have. The hope is to have targeted intervention. We're not there today. And with that, thank you.